Hello, Professor.
Hello, Scott. How are you today?
Good, good.
Good,
How
good,
are
good.
you?
I don't know. I'm feeling sick today for some reason.
Mm,
I have kind of a headache,
yeah.
my full head. No sneezing, no coughing, but I have a sore throat, like lasting now for almost 2 weeks. You know, coming and going. It is not like persisting. So, and I don't know. It's maybe allergy or something. But no fever, nothing, But the headache today is kind of a little bit tough.
Yeah.
Yeah.
Hard to concentrate.
Now the problem is now anybody, you get a headache or some like sore throat or anything like this, or maybe some sneezing. You will go and run to a conclusion and say, "Oh, I'm having COVID." You
Hmm.
know, it's an uncertain situation. You know.
Yeah.
Everybody is kind of panicking.
Hmm.
Yeah, man, we'll see what will happen, It's a situation which we need to deal with, unfortunately.
Yeah.
Okay, so I can see only you and another student with me. Hello,
I I am here, sir. How are you,
how are you
sir?
doing today? You good? We missed you last week.
Yeah, but I was here.
Yeah.
I was with you.
Oh, you was with me? Oh, okay, that's
Yeah,
good. That's good.
yeah.
Okay, so let's do this. Let's divide the talk today into two major topics. One of them will be the going quickly to the identification of the unknown bacteria for the lab, and
Mhm,
then I have two chapters, 19 and 20. Chapter 19 is for gram-positive bacteria, and chapter 19 for gram-negative bacteria. There are some selected topics that I'd like to go through with you, uh, unless you have other question for me. Okay. So before I start, anyone had any question for me for any other thing, like for the lab, for lecture, for exams, or anything?
I have one question about the this assignment, our
Yeah,
assignment for the lab only. Yeah,
the
the
lab,
difference.
the
Yes,
lab.
in the lab.
Okay,
Yes,
yeah. So what is your
uh,
what is your question?
that's uh, that I want to know the difference between culture and
Culturing and isolation.
isolation, and which one is comes first? Isolation is comes first, or culture is comes first? This is my question.
It's a good question.
I
Okay,
know,
so
so
if I
maybe it
gave
is so
yeah
simple for others.
if I give you like a soil sample,
Uh,
okay,
uh-huh,
and I say, okay, go ahead and isolate whatever microorganism on this soil sample. So
uh-huh.
what you will do first? So
So I
imagine
saw.
this: I give you a soil sample,
Mhm,
and I give you like say some tryptic soy broth, and I give you an incubator and said, okay, now you have a soil sample. I'd like to isolate some microorganism which inhabiting this particular soil. So what you do first?
uh,
Your first step will
culture.
be culturing. Yes,
Yeah,
exactly.
I will go.
Yeah,
Yeah,
yeah.
I
You
will
need to
culture it.
culture. Yeah, you need to culture whatever you have
Uh-huh.
on the
To
on the soil sample first, because
Yeah.
you need to grow them and you need to increase their numbers to a point that you will be able
Uh-huh.
to see them. And
Uh-huh.
then when you grow them, say in nutrient broth, it will be hard to isolate anything because
Uh,
it will be a mixed culture. Okay. Now you
yes.
need to take from this mixed culture and do plating on solid media so you can
Uh,
see different kind of colonies.
yeah.
Whatever it's mold, it is fungi, it is bacteria, and
Uh-huh.
now you will be able to isolate from this culture.
Okay.
Yeah.
Then culture
Yeah, you need
comes
to culture.
first.
Yeah, you need to culture first and then isolate.
Okay.
Okay.
So thank you very much.
Or if I give you an isolate, say I give you a plate which
Mhm,
already has isolated culture, isolated bacteria, say a plate which has maybe 10 different kind of microorganism. Then
mhm.
you take from this isolate and you do culture, you know. So it's a
Yeah.
process of overlapping depend on what you need to do. So if I give you a sample of soil, you need to do culture first and then isolation. But if you have the
Okay.
isolates already in your hand, then you can
Mhm.
go ahead and do culture directly. This is,
Okay.
yeah, basically. Okay.
Okay. Okay. Thank you.
And culture doesn't mean that you have a single organism. Culture, you have a mixture. And only you will be able to culture single organism when you have them separated from each other. Then you can
Oh,
make pure culture. So there is a difference between a culture, initial culture, and pure culture. Initial
oh.
culture meaning whatever you have in the soil sample.
Mhm.
Okay. Pure
Initial
culture
culture.
is initial. Pure meaning it's a single organism. So I give you like a plate with E. coli, and you can take a single colony from this plate, and you
Mhm.
can make a pure E. coli culture from this.
Okay.
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 Yeah. Yeah.
Yeah.
Yeah.
This is my question.
Okay.
Yeah.
No problem. Yeah. Anything
Yeah.
else? Yeah.
Thank you, sir.
No problem. So anything? Any question for me before we start?
Okay.
Okay. So. This is the lab that you are going to deal with this time is a bacteria identification lab, and as I mentioned before, maybe I didn't get the chance to post any video, so I will go through this quickly. I think the due date for this is going to be tomorrow, July 14, I believe, right?
Yeah.
Yeah.
Mhm.
So it is rather I'd like to go through this quickly. So whoever is still confusing, hopefully this will find some answer. So I provided the class with three different three different unknown, and assigned groups. Okay, so you'll see maybe 6 or 7 students, and they have unknown 1, 7 students unknown 2, 6 students, for example, unknown 3. It doesn't mean that you need to work together. No, you don't need to work together in this. This is individual assignment, because I don't have, if the class is 24 students, I don't have 24 unknown. I have only 3 unknown. So you will share this. It doesn't mean that you need to work together or communicate or talk. It's completely individual assignment. Those unknown came from the family Enterobacteriaceae. And family Enterobacteriaceae is a big, huge family in bacteria. And it has lots of genera, and those are the major genera under this family. E. coli, Salmonella, Shigella, Yersinia, Enterobacters, and so on. So those are under the same family sharing some common features. Those common features are the following. They are all gram-negative. They are all short rods, or we call it bacilli. Some of them we call it coxobacilli. Coxobacilli, it is not cocci, it is not spherical, not completely rods. Okay, so since they are very short, so under the microscope you will see them. You don't know what is this. Is it spherical cocci or is it rods? Because it's coxobacilli. Okay, this is one of their features, and they are all gram negative. Gram negative meaning they are staining red with the after adding the counter stain safranin. They are all gram negative. Majority of them are motile, remove. Okay, and the way they move with flagella, and the arrangement of the flagella, and most of them is kind of peritrichous. When I say peritrichous, meaning it is surrounding the whole bacteria. Okay, so the other thing which is important about those, they are non-spore former, so they cannot make any spore. So under harsh condition, they will die. Okay, they cannot tolerate high temperatures, they cannot tolerate high salt, change in pH, and so on. oxygen requirement, they are facultative anaerobic. Facultative anaerobic, meaning that they don't care if oxygen is present or absent. So if you grow them like under 3 different conditions of oxygen, like we call it ambient air, which is the oxygen concentration, normal oxygen concentration in the atmosphere, which is 21%, they grow very well. If you put them in a candle jar, which a jar that has a candle, burning candle, to eliminate some of the oxygen. So I mean here, less oxygen, so it will be significantly less than 21%, maybe roughly 10% oxygen concentration. They also can grow. Or if you eliminate oxygen completely, they also can grow. So we call this facultative anaerobic, okay? So your job is basically to take that specified unknown and you need to identify it for me at first of all, if you will be able to get the genus, and then it will be great if you get also the species. So there are many biochemical tests that you need to run to do this kind of testing, okay? Before I go to this, they all are catalase positive. So if you treat the culture with hydrogen peroxide, you will see oxygen release, which give you this kind of bubble. They are all enterobacteriaceae. They are all oxidase minus. So the oxidase negative, and as I mentioned, they are motile, as you can see in the tetrazolium chloride broth. Okay, tetrazolium chloride deep agar. Okay, so this is a chart here for a very, very important test that you need to follow and identify your unknown based on the data, okay? And this is very important because in your practical, second practical, will give you some of this media, the names you can see, and I will ask you what is this media or what is this stands for. If I say SIM, you need to tell me that this is a media which is designed to test for hydrogen sulfide production, indole production, and also for motility. Okay, what is the IMVIC test stands for? It is a big, huge biochemical test which tests for indole production, which tests for the ability of the organism to ferment glucose and produce acid. So M here stands for methyl red, and Voges Prosker VI, it's another test for glucose fermentation, but we're not looking for acid. We're looking for something different. I will show you in a minute. And the C stands for citrate utilization, the ability of your bacteria to utilize citrate as a sole carbon source. When I say sole carbon source, I mean the media. When I prepare the media, I make sure that the only carbon available will come from citrate. I'm forcing the bacteria to use it. If the bacteria have the enzymes and the genes responsible for those enzymes, so to utilize citrate, they will grow. If they don't have this machinery, they will not, okay? So for those tests, also in your practical, you need to know the indicator used, because most of those tests, you need to add an indicator to be able to interpret the data, okay? For example, if I'm testing for hydrogen sulfide production, the bacteria, if the bacteria is capable of utilizing sodium thiosulfate, for example, it will, if it produces hydrogen sulfide is colorless. So there should be a way to be able to detect it. And this is how to detect the hydrogen sulfide. Ferric ammonium sulfate, which will react with hydrogen sulfide. I will give you a very black color like this. So this is a media here without, say this is your initial media, yellow. You grow your organism. After you grow it, it's still yellow. In order to be able to detect hydrogen sulfide, you need to add ferric ammonium sulfate and look for this color development. If it is still the same, it means that the bacteria is negative. They are not producing it. The other thing is the ability to produce indole ring. What is indole? It's a byproduct from tryptophan. Tryptophan is an amino acid. Some bacteria, they have enzymes, tryptophanase, which break down tryptophan and produce indole. Again, you should add something to be able to detect indole. And this is what we call Kovac reagent. So if you add Kovac reagent and you have indole, it will give you this very nice red color at the top. If there is no, it will stay the same. Okay, so this is a very important test. What I said here about glucose fermentation, and for glucose fermentation, you are looking for two different products: acid production and the production of what we call acetone, or a neutral product called acetone. Okay, so you grow one tube only. So you take one tube broth that has glucose as a carbon source and other ingredients. No indicator, nothing. You grow the bacteria, and probably 24, 48 hours later, you take aliquot from this, okay, in a test tube which is clean, okay? And for the rest of the broth, you add methyl red. Methyl red is a pH indicator. Remember, two major pH indicator, one of them is phenol red and one of them is methyl red, and they are opposite. So phenol red, it's red, and if the pH decrease toward the acidity, it will turn yellow. Methyl red, on the other hand, is red, and it will stay red under acidic condition. It will only turn yellow under basic condition. So they are opposite. You don't need for me to confuse those. They are opposite. So if you take aliquot, if you add like methyl red to your culture, and the culture is basically change color to red, it means that it produces acid. If the color stays the same, okay, that is meaning they are not producing acid. Like in this culture here, they added methyl red, and as you can see what happened here, it turned the media is yellow. So if the media stay yellow as is, so it means that this is negative. There is no acid production. But if you add methyl red to the culture and take the color of the methyl red, it means it's acidic. It means it produces acid. For the second test, you take the aliquot and you add your reagent, okay? And the reagent, you need to know the name of the reagent. Those are Barrett A and Barrett B. You need to add those to the culture and mix and wait. This kind of reaction needs oxygen, meaning that you don't need to close the tube. You need to make the tube, to keep the tube open to the air. So the color is going to develop. If the bacteria develop this kind of color, it means that yes, they were capable of utilizing glucose and producing acetone. So you need to remember the name, acetone, okay? You need to remember the reagent used for this test, for Voges-Proctor testing, okay? It is very important. You don't need to worry about the chemical composition or chemical structure of acetone. You don't need to worry about this, okay? So any questions so far?
No.
Okay. This is the citrate testing. It is important too, and this is you need to know in this what is the name of the test? Simon citrate media. Simon citrate media. Simon citrate media. It has only citrate as the only carbon source. Simon citrate media is green, and when you start culture or putting your bacteria, it's green. Okay. It has an indicator, and that indicator that make it green, it is called bromthiamyl blue. So this is another confusion. We said, okay, the media is green and the name of the indicator is blue, because it will develop to blue color later if the bacteria is capable of utilizing citrate. Okay, so this is before incubation, and this is before culturing, this is after culturing. If the media turn to nice blue color like that, that means that it's citrate positive, okay? The other test, which is so important, and I received like a few emails about that particular test. You are here using testing for 3 important decarboxylases: arginine, decarboxylase, lysine, and ornithine. Okay, those are enzymes, and the name decarboxylase means an enzyme that has the ability to remove the carboxylic group from those amino acids. So the media itself, it has an indicator, okay, and that indicator is bromcresol purple. And before incubation, the media is purple, okay? So the media is purple before adding any bacteria due to the presence of bromcresol purple on it. If the bacteria is capable of decarboxylation and removing of carboxylic group, okay, that the end product is going to be basic. So arginine or lysine or ornithine, because remember those amino acids are unique because they have 1 carboxylic group and they have 2 amino group. If you look at their structure, we call those basic amino acids. They have 1 carboxylic and 2 amino group. So removing on one of those carboxylic group will make them more basic. If it is the case, the bromcresol purple will stay the same. It's not going to change color. Bromcresol purple will change color only to yellow if there is no decarboxylation is happening. So remember that the media before is purple. If it stays purple after incubation, it is positive. If it turns yellow, it is negative. This is very confusing for some students because they believe that the media will start with a purple media, and if it turns to yellow, then that will be a positive reaction. No. Okay? And you need to know the name of the indicator. Bromcresol purple. So there are bromthymol blue for Simon citrate and bromcresol purple for the arginine, lysine, ornithine decarboxylase testing. Also, there is another test for phenylalanine deaminase. You are searching, you're looking for this an enzyme which remove the amino group from phenylalanine. And the way you detect this, you add ferric chloride. Okay, if ferric chloride react with if the phenylalanine lose the amino group, it will be acidic. It will react with ferric chloride, and it will give you this very nice avocado green color. Very, very nice. So if you see this, you would say that your unknown can deaminase, or it has a deaminase that can remove amino group from the phenylalanine amino acid. Those are 2 important tests. So here, what you will have, you have the media before inoculation and after. So if this is a media that you are testing, say for hydrogen sulfide production and indole production, if you add the reagent, see here, after incubation with reagent, meaning you add the reagent, no color change. So it is hydrogen sulfide minus. Indole ring, no red color. So it is minus. So this is the way how you interpret this kind of data. Okay. So after you read all those tests, you need to go here in this slide, and that will tell you, how to. It give you some hints, not complete, but it will give you some direction. Okay, how to identify your unknown. The first test and the most important in Enterobacteriaceae, okay, we call it the lactose fermentation, because that lactose fermentation differentiate or separate Enterobacteriaceae into 2 large groups. One of them, we call it the coliform bacteria, which is mainly inhibiting or inhabiting your colon in the large intestine. So this is why we call them coliform. Coliform is capable of fermenting lactose and producing acid, okay? And the non-coliform, which is not capable of fermenting lactose. So go back here and look at your lactose. In this experiment, you have 4 different types of sugar, and you are testing your organism and see if it can use those different kind of sugar. So you are doing, we call it carbohydrate broth. The media, it has glucose, it has lactose, sucrose, and mannitol separate. Each tube has a specific sugar. And also, you need to know the name of this inverted tube here. There is a small tube, inverted tube, called Durham tube, D-U-R-H-A-M, Durham tube. This tube is made to capture any gas during the fermentation process. You have an indicator in the media. The media is red. It has phenol red. So it is not methyl red, it is phenol red. If the bacteria is capable of fermenting the sugar, the media will turn to yellow. If the bacteria is capable of producing gas, gas will be accumulated, as you can see. So in this case, you have bacterium that is capable of producing gas and producing gas and changing and producing acid. Okay, so you go back and this is, as I mentioned, an important test to differentiate between coliform and non-coliform. So if you look at your test and you see it can ferment lactose, then the second thing that you need to look for is going to be hydrogen sulfide production, okay? And then as soon as you see hydrogen sulfide production, it will, if it gives you positive, you will say, okay, this is Citrobacter. So that will take Citrobacter from the equation. If it is negative, now you need to look for another test to be able to separate and so on. You will see Citrobacter here and Citrobacter here. What does this mean? It means that there is some variants of Citrobacter that cannot produce hydrogen sulfide, okay? So it doesn't mean that every single Citrobacter will produce hydrogen sulfide. The majority will do, but still some cannot. But you will be able to capture them later if you do different kind of reaction, okay? So from those, the class have only 3 unknown. In your job to tell me your unknown. As soon as you do this, you have an extra. This one is more informative, kind of more clear. Other biochemical testing you need to look for. So you will be capable of identifying your unknown to the species level. This one is kind of hard to read, you know, but this is from Berg's manual page. In the lab, in the real lab, we used to have Berg's manual sitting on the bench, so you can look at it. And, you know, for other, if you have any question for your unknown. As soon as you finish and identify, you need to provide me with this sheet, okay? So it has the isolate number that I gave you. You need to check what test, positive, minus, and then write down probably this organism is whatever. And then as soon as you get comfortable with this and you're sure, you write like 2 to 3 page essay about that particular unknown, a medical importance for it. So any question for me? And one test here. Let me see here. Sorry about the noise in the background. My kids are crazy. Okay, so there is a gelatin hydrolysis. It's a very important test, and with the way we interpret gelatin hydrolysis, gelatin, as you know, is basically protein. And if you grow your organism and your organism is capable of hydrolyzing gelatin, what is going to happen? That gelatin will lose its property and will be liquid. So you have gelatin and you have bacteria, you inoculate and you incubate maybe for a couple of days, and then you take it from the incubator. Everything is going to be liquid, but you need to make sure if the organism was capable of utilizing gelatin or no. So you need to take it and put it in the fridge, like for maybe 1 hour or so, or put it on ice. If the gelatin is still intact, it will be solid. It will remain solid. If the gelatin is already degraded by the bacteria and you take it from the fridge, it will be liquid. This is the way how you interpret the gelatin. So in this case, if you look at this gelatin hydrolysis, it's gelatin hydrolysis is negative. Why negative? Because gelatin is still intact and make the media kind of solid. Okay? Is another also some students send about this particular test. So any question so far for me?
I have a quick question. You're on, like, I think test 1 or the first isolate, and I emailed you about it, but the urease test, however you say
I think
that.
I responded to
Yeah,
you.
yeah.
Urease test.
So, yeah, so the light pink, if you see that and it's just like the uninoculated, if it is light pink, then that's just a weak positive, and that will always be positive on your results or on your.
Yeah, for the urea test, basically the media is kind of, when you see it in the lab, the media has this kind of color. It's not pink. It's a shadow of, I'm not as good in coloring. I don't know very much, a problem. But the media, the color before inoculation is not pink. It has no degree of pink at all. Okay, it is kind of not yellow too, but it has a color which is not yellow, not pink. And the way you interpret the data, you will see if it is very, very good and very positive, you'll see beautiful pink color in the media. Okay. But if it is weak, so they can like hydrolyze urea, then it will give you some kind of shadow of pink, like here. So if you look at this, you don't say it's negative. No, it is not. It is positive, but it is weak positive. It is weak positive. I hope I have another slide that show you how. Let me see here if I have another. Test for this. Okay, bear with me, please. This is here. Let me show this other one here. Okay, let me stop share and share with you another presentation, okay? So let me share with you this one. Can you see this one?
Yes.
Yeah, this is what we call a positive, very positive.
Okay, that makes.
Okay, this is the way it looks like. Okay, but if it is the one that I saw you a little bit earlier, no, it is kind of weak. It is positive, yes, but it is weak.
Okay.
Okay.
And just for example, seeing as we're not actually like able to do these in a lab, on the like one of the charts, it says plus or minus usual reaction or D for different. What would be like an example for a different reaction?
Different reaction meaning that if you have a variant of that genus. So say I'm talking about Escherichia or Salmonella, for example. So Salmonella, some Salmonella will give you like a positive, and some salmonella will be different, give you a different colorization, different end product, different interpretation at all. It is not going to be the same. Those, we call those variants within the same genus. Okay, and
Okay.
I give you an example here. This is a very, very authentic example about Citrobacter. Majority of Citrobacter can produce hydrogen sulfide, okay? But
Mhm.
there's still some Citrobacter cannot produce it. So when you do when you do the identification and you go to hydrogen sulfide minus or plus, if it is plus, you take Citrobacter, it will be Citrobacter here. But doesn't mean that all Citrobacter will give you the same feature, because if you do the test more, some other Citrobacter will come later, although they are minus for hydrogen sulfide, but still variants from the genus. Some mutations, stuff like that.
Okay.
This is why the biochemical testing is not accurate 100% for identification. This is why genetic testing is very, very important. Basically, you look at the gene level and the sequence
Okay.
for identification. But in this, it is very, it's more than 95% or 97% accurate, all this biochemical data. But if you get so confused about an identification, then the DNA will be the judge, the final judge.
Okay.
Okay, any other question for me? Any question for me for this lab?
I don't have any.
Okay, so good. So let's now, we have those two chapters here, and let's do this first. Gram positive. Do you have already those two chapters, 19 and 20? Those two chapters, chapter 19 is focusing on the pathogenic gram-positive bacteria. Chapter 20 is focusing on gram-negative bacteria. Okay, and what you need to know from this, for gram-positive bacteria, like probably next week or the week after, I think next week, we're going to start two major genera under gram-positive, and those are Staphylococcus and Streptococcus. Okay, those are two gram-positive bacteria, two major genera. I will give you like also for like an assignment for how to isolate and identify. Okay, we did this again in the lab, but unfortunately we will not be able to do it. So there will be like step by step how to isolate it and how to identify what you have here. So these are some features of Staphylococcus, for example. And when they say cocci, you know that they are circular. Okay. When I say staph, they are the arrangement here. The arrangement of those cocci is kind of clustering, as you can see. It's like grape, okay, cluster of bacteria. Those specific group are non-spore formers, so they don't produce spore. Also, they are facultative anaerobic, like Enterobacteriaceae, okay? They are non-motile, they are not movable, they can move. They have salt tolerance, halodurant. They are salt tolerance, meaning that if you, by any chance, you have a high salt in the media, they can grow, okay? But they don't like it. They are not like halophilic. Halophilic, meaning they like salt. Salt is essential for them to grow. So if you have a halophilic bacteria and you grow it like in normal nutrient broth or tryptic soy agar, for example, with 0.5% sodium chloride, they are not going to grow. Okay, you need to add maybe 10%, maybe 12.5%, 15% salt for those bacteria to grow. Staph, they can grow up to 10%. They can tolerate up to 10%, but it is not requirement because it's halodurant. Okay. The other thing, there are two major species under this genus, Staphylococcus aureus and Staphylococcus epidermidis, okay? And those are epidermidis, from the name, it lives in your skin, and they are opportunistic pathogen. When you say opportunistic, what does the word opportunistic mean? Opportunistic mean if they get the opportunity to infect and cause disease, they will. They are not going to hesitate, okay? But they can live with you as soon as you have a very nice, your skin is nice and clean, no scratch, no burn, nothing. They are in your skin and they are fine. But as soon as they get the opportunity to to invade, or you have a scratch, you have a wound in your skin, a burning, and it got contaminated with those evil guys, then that will be the real problem. They can do lots of damage. So like Staph aureus, for example, can do lots of damage. And the reason for this, we call it pathogenicity. They have tactics. They have very important thing that they can do to escape and cause lots of disease without getting harmed. One of them is their ability to escape your immune system, especially from phagocytosis. And I will show you a very important slide in a minute. Okay, phagocytosis, which is very crucial step in your body to eliminate those invaders. They can escape from this. They produce a cocktail of enzymes and toxins that help them invade the host. Okay. One of the things that they are producing called protein A. Okay. Protein A, this is in the surface of the cell. So what does protein A do? I will show in a minute. Protein A from Staphylococcus, it has the ability to bind to immunoglobulin. And when they do this, they prevent a phagocytosis, antibody-mediated phagocytosis. When the macrophage trying to identify like a foreign body or a foreign cell, that foreign cell need to be tagged. It has tag, like flag. And the flag in this case is immunoglobulin G, an antibody that you produce. That will tell the macrophage to go ahead and clear the body from this tagged organism or cell that is coated with immunoglobulin G. What Staph do? They produce a protein that bind to that immunoglobulin and preventing a very crucial phagocytosis step. We call it opsonization. Opsonization here meaning that the macrophage will be capable of taking the foreign cell if they are tagged with antibody like this. Protein A prevent this from happening. I will tell you in a minute how this happens. It also produces some important enzymes like coagulase. A coagulase, and this is one of the important tests that can differentiate to take Staphylococcus aureus out from other staph. Coagulase here is an enzyme which is converting fibrinogen to fibrin in your blood. So they make clots basically. Okay, they convert fibrinogen, which is in a liquid format, to fibrin, which is solid, and they do this to like hide. So they induce clotting, blood clotting, to hide inside those blood clots, and the phagocytic cell will not be able to see them. Okay. Very, 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 very dangerous kind of microorganism. And then they have also another enzyme. When it is safe for them to come out from those clots, they break down these clots and release the bacteria again so they can do their job. Okay. Here is in phagocytosis, there is two processes. One of them we call it complement mediated, and one of them we call it antibody mediated. Okay, so complement mediated meaning that there are some complement proteins in your immune system which are essential for the process of phagocytosis to happen. So you imagine you have a bacterium, okay? And the bacterium, you know, there will be some complement protein secreted from immune system, will bind to the bacterium and will flag it, make it like flag, like a sign that this is ready to be eaten, okay? So a phagocytic cell, they have receptors for those complement proteins on their surface that can interact with the complement. If this is the case, this kind of interaction will lead to phagocytosis. In no time, the phagosome, including the bacteria, will be inside the macrophage, and they will be degraded into pieces, into amino acids, into nucleotides, whatever. Okay, this is, we call it complement mediated. This is how your immune system recognizes foreign cells. Also, similarly, there is also what we call antibody mediated. Antibody, where there is you have the bacterium and you have specific antigen on the bacteria, so there is antibodies that can interact. The antibody, if you recall, it has several chain. One of them, we call it the Fc fragment at the end of the antibody, Fc fragment. And that Fc fragment is recognizable by a receptor in the macrophage. So if an antibody interact with the bacteria, then the whole thing is going to be taken in via the Fc receptor, okay? This is normal cases. This is no staph here. So how staph manage to escape from this, okay? So before I move to the next slide, because I selected those because I believe those are the most maybe kind of hard to understand, okay? So any question for this before I move to the next slide?
No questions.
Okay, so here, how a staph is managed to escape. So again, you imagine you have, this is a macrophage, and they have receptors, okay, on their surface, and this is your bacteria. So this is your staph here. So the staph, they have protein A. Protein A. This is, they are coated with this. This is a bacterial protein. Protein A is a bacterial protein, okay? So protein A, if the macrophage is like trying very much, you know, to reach to the staph to capture and take it, okay? So the macrophage, they have receptors, okay? One of the receptor is a receptor for the Fc fragment of the antibody. So if the antibody will come and start, you know, like bind to a specific protein surface on the staph, protein A is going to bind to the Fc fragment of the antibody. When it does that, bind to it, it will do some sort of steric hindrance. It will prevent the receptor on the macrophage to recognize it. It is blocking it, as you can see. So when it does so, now that interaction between the receptor on the macrophage and the antibody on the staph is not going to happen. Why? Because you have like protein A is blocking this kind of interaction. So that will prevent the phagocytosis process. So this is the way they do to escape from this. One way to escape from the immune system is to produce the protein A, which is preventing interaction with the receptor on the macrophage, preventing the phagocytosis. Or they also produce the blood clots that will surround them and prevent macrophage from identifying the bacteria. Or some of them, they produce capsule, polysaccharide on the surface that also will prevent phagocytosis from happening. So they are kind of smart and dangerous kind of bacteria at the same time. Okay, you have any question for this, anybody? I know maybe it seems complicated, but the bottom line here, you need to know there is a process called osponization. Osponization here, meaning that the pathogen should be coated and identified with either complement, protein, or antibody to be phagocytosed. Okay? Staph inhibit the process because they produce a bacterial protein which prevent this kind of interaction between the antibody and the antibody receptor on the macrophage. Yeah, please ask me if you have any question, because I think asking question will open the door for discussion, and if there's something not kind of like confusing, that probably will make it more easy to understand.
So, Professor, even though the pathogen produces the protein A
Yes,
to inhibit the kind of real smooth adhesion to the
the antibody
phagocyte,
receptor, yes.
but it still is absorbed, even though it kind of makes it harder.
Absorbed by what?
The phagosome.
No, it will not.
It will
Because
not.
as you can see here, I have this nice yellow, this is in biochemistry, meaning
Yeah.
blocking.
Oh, okay.
It will block, it is blocking. So it's not going to happen. It is not going to happen.
And so it just kind of pushes back out.
Exactly. It will, the whole thing is not the receptor
Yeah.
on the macrophage is not going to recognize
Yeah.
the antibody, because the antibody, the Fc chain of the antibody is blocked with protein A.
Yeah.
So it's like a strict hindrance. It's like something in between that prevents this kind of protein-protein interaction.
Mhm.
And without this kind of interaction, no phagocytosis will happen.
Mhm.
Yeah.
It's like when we were studying the active sites of enzymes.
Exactly.
Yeah.
Absolutely. Yes. When you have an enzyme and you have like inhibitors, you know,
Mhm.
those like active site, like
Yeah.
inhibitors or allosteric inhibitors or the competitive inhibitor, it's a competitive inhibitor
Yeah.
because it competes, you know, with the binding, you know. So now you have the Fc of the antibody, you know, and this one compete, it prevented from binding to its receptor. So it's a competition here.
Yeah.
You know, so and based on the concentration of this protein A, it's lots of factor, but the bottom line is a very important process to clear body from infection
Mhm.
is not going to happen.
Yeah.
It is a problem.
Yeah. I had a friend that got staph, I think from a gym. I don't know. He was playing basketball or something like that, and
Who
he
got injured, like
he
some
got a
injury?
cut or
Oh yeah, man,
when he
yes.
was playing, and
Yeah.
it really messed him up.
Yes, yes, I'm telling you, it's just a small cut burning, and those kind of staph, they are opportunistic. They are living in your skin, in your hair, on your body, and inside your nose, inside your in your throat. Like strep, strep is inhabiting your throat. Staph is inhabiting your nostrils. You know, the upper part of your nostrils. But thanks, because we have other bacteria which is competing with them. You know what I mean? So it's always competition. And this is a problem when you have like say infection and you take antibiotic, and if you have one of those staph which is resistant to antibiotic, so the antibiotic will kill other bacteria, and then the staph will flourish. There is nothing can stop it, so it will start, you know, it's bad stuff. Okay.
Yeah.
These some of the enzymes they mentioned that they can produce. Okay, so this is the enzyme hyaluronidase. This is the one that break down the the uh, kind of call it hyaluronic acid and enable the bacteria to spread between cells. It is kind of hyaluronic acid is like a protein. Sorry, it's like a component. It's a connective tissue between cells, and this is the way how staph can spread. So they break down this kind of hyaluronic acid in your connective tissue between cells and make the cell loose, and the bacteria can spread easy between cells. It have the staphylokinase, and this is again that dissolve the fibrin that around the clot. So the bacteria form clotting with coagulase to escape, and then when things start to calm down, they produce another enzyme, staphylokinase, that break down this clot and releasing the bacteria so they can do their infection. Okay, so they are basically the bottom line: bacteria which is loaded with enzymes, which is loaded with proteins, and different kind of way to escape from the immune system. This is why they make them so pathogenic. You know about MRSA, right? MRSA, when they produce some enzymes like beta-lactamase, remember this is an antibiotic here. Say this is methicillin, okay? And this is the lactam ring, if you remember. So one way for them to escape is to produce beta-lactamase that can degrade or break down this lactam ring, beta-lactam ring, and that makes the antibiotic ineffective. It can't do anything. And this is like a methicillin-resistant Staphylococcus MRSA aureus. Okay. What else here? The Clostridium is a very, very nasty gram-positive. Clostridium is different from staph because Clostridium is rods, so they are bacilli, not cocci, and they are spore form, and this is a big problem. Okay, they are spore form. Now the scientists will able to use like some proteins from Clostridium, the botulinum toxin. Okay, botulism is a very, very, very dangerous toxin, and now they can even produce it and purify it from the bacteria and use it like some products like Botox, if you heard about that. So Botox, they can use it to inject, and all this kind of muscles which is contracting, as you can see, they can do some kind of relaxation of the muscles. So you can see this, okay? So this is a good thing about them, but they are bad. And how I have one slide I need to show here. What about botulinum toxin? How botulinum toxin work? Okay, it's kind of a complicated slide, but what you need to know from this. So the Clostridium, they are first of all spore former, and the other thing which is so unique about them are anaerobic. Anaerobic meaning they are strict anaerobe. Oxygen will kill them, okay? Oxygen will kill them, so they can't grow in oxygen at all. And their way of killing and infecting people is producing toxins. One of them is this botulinum toxin. And the botulinum toxin is basically prevent muscle contraction. So it causes some sort of muscle paralysis, okay? Muscle paralysis, how? Normally, you know that there is something, there is the muscle cell is always in contact with a neuron, a nerve, okay? And there is a space between the muscle cell and the neuron, okay? And we call this is the space where we call the area, the whole thing here, we call it the neuromuscular junction. So it's a connection between the muscle and neuron, okay? And the way the muscle contract is basically, or stimulated, is basically comes from the neurons surrounding it. There is a space here between the muscle cell and the neuron. So what happened is the neurotransmitters, like acetylcholine, for example, comes from the neuron cells in vesicles, okay? And it releases its content in this junction between the muscle and the nerve. And those neurotransmitters will bind to the receptors, those acetylcholine receptors on the muscle cell, and that will lead to contraction. So this is how the muscle contract. So muscle contract via release of those neurotransmitters, okay? But for the vesicle that carry the neurotransmitter to work and to to be like trafficking to the membrane, there are some proteins essential for this process, okay? Like syntaxin, SNAP25. Those are proteins. They are present on the membrane of the neuron. It recognizes the vesicle and allow those to release. This is normally. What if, for any reason, you was eating some food, and it happens a lot, especially in my country, back in the years, where they eat salted fish, and if by any chance salted fish is kind of contaminated with the spores from Clostridium botulinum, it will be huge. It can be fatal. The bacteria can grow in the intestine and then release toxins, okay? And those are the botulinum toxins. For some reason, those toxins, they know how to get on the junction between the neurons and the muscle, okay? Very, very unique. And they have receptors. They have those toxins, they have their own receptors. They take advantage of some proteins on the host that they can bind to. And as soon as they bind, that will mediate endocytosis. So in no time, the botulinum toxin will be inside the cell, inside the neuron now, okay? Inside the neuron. Botulinum toxin is made of two subunits, as you can tell. One of them is the catalytic subunit. So one of them is the delivery, which you deliver, and the other one is the catalytic. That catalytic, what is its function? Its function to break down those important proteins. Okay, break down those important proteins. So now you have neurotransmitters inside, and for those neurotransmitters to be released, they need to bind to those receptors. Now there is no receptors. So what is going to happen? Neurotransmitters are not going to be released. Okay, so the muscle is not going to contract. So the muscle will be like a paralyzed, especially if you are talking about the muscle of the diaphragm. This muscle is so important for breathing, as you know. Moving this muscle will move your lung. So if for any reason that muscle get paralyzed, you can breathe. You will suffocate, and you person will die. So this is how those toxins are so dangerous, how they work. Here it says 100 nanogram of this pure toxin can kill an average human. 100 nanogram, you know, a nanogram is 10 to the negative 9 of a gram. So it is 1 billionth of a gram. You imagine a gram, 1 billionth of this is enough to kill a human. So toxic, so toxic. And it's a problem because they are spore former, and you know, spores are so tough to be treated. So if you have canned food contaminated with this bacteria, it will be a problem.
Can the endospore go in hibernation and you can be well, and then it can come back after a while?
Okay, now that the endospore, what they like, as soon as they found the condition in the environment and around them is wet and nice and nutrients, they sense this kind of condition, so they germinate. And as soon as they germinate, they produce the toxin. And that toxin can get its way to the blood, and it can move straight to the neuromuscular junction and to the neurons where it starts its activity. And it's an enzyme, basically, main job to break down. The thing that I don't understand, why they do this. I mean, what is the benefits they will get from preventing muscle contraction? I was trying very much to search and see what is the idea for this bacteria to prevent muscle contraction, how this will help. Because you know that those bacteria or microorganisms, what they do, they need to propagate and increase in numbers, basically. It's their job, like viruses, okay? So their job is to terminate the host, to get rid of it. So till now, I don't know why muscle contraction is going to help them, you know. but it's a tough organism. The brother of Clostridium botulinum is Clostridium tetani. It's another nasty Clostridium. Tetani is also rod spore former, okay? And it produces specific kind of toxin, we call it tetanus toxin, tetanus, okay? And now how tetanus work? It's opposite. So botulinum is paralyzed the muscle. It prevent the muscle from contraction. Tetani or tetanus, on the other hand, will allow an overexciting muscle contraction. Okay, overexciting muscle contraction. You know those lock jaws things, flexation to the point that you can crush the bone from the muscle when the muscle like contracts so hard. So this is the tetanus work this way. Again, same thing. You have here, but here's the thing. You have, this is a motor neuron which is connecting to the muscle. Okay, tetanus, Clostridium botulinum toxin work here. Okay. Okay, but the tetanus work in a different area. So here you have, this is the motor neurons, and you have two different kind of neurons. One of them we call it the stimulating neuron, and the other one is inhibitory neurons, and they are working together. Okay, one of the neuron is releasing the neurotransmitters to like make contraction for the muscles, and the other neuron inhibiting this kind of contraction, so it will be relaxation. So they are working together for the muscle movement, okay? Normally, under the infection with tetani or tetanus, what's going to happen? The tetanus toxin is going basically to bind, it has some specific receptor on the inhibitory neurons. And when it does so, it prevents the movement of those vesicles with the inhibitory transmitters to be in. So always it will be stimulating. Always stimulation is going to the muscle and no inhibition because it blocks it, okay? So that will lead to this kind of contraction, as you can see in the picture. So it's opposite. This is why there was, some people said, what if somebody get infected with botulinum to add, to treat them with tetanus toxin? Because botulinum is paralyzing the muscle. Tetanus, on the other hand, is contracting the muscle. So adjusting the dose for tetanus, you will basically prevent this from happening, and it will only, the stimulating neurons will more stimulating neurons will work, okay? So that will allow this kind of situation to be resolved. It's kind of complicated, huh?
Yeah, a question. So when you visit the doctor, why are they so adamant about asking about tetanus shots all the time?
Yeah, tetanus shot is very important because when you get this, you are, it's very good to have some antibodies, memory for the tetanus toxin, okay? Because remember how those bacteria can, how you get those bacteria. First of all, those bacteria are anaerobic, you know that, they are anaerobic. So you will not see them in your skin, you will not see them in your hair, okay? They like to be completely away from oxygen. So they are hidden, say, in the soil, spores, soil, deep in the soil, okay? So say, for example, you was playing without any shoes outside, and there is a nail or some rock or something that makes some like wound in your feet, for example, deep one, and for some reason it got contaminated with soil, with dust, soil, and that soil was loaded with tetanus, the Bacillus Clostridium tetani. So Clostridium tetani, the very thing they will do, they will go deeply inside and where the oxygen concentration is very low or no at all, and they will start germinate and produce the toxin. So if you have shot for tetanus toxin, now you have antibodies. So those antibodies are going to recognize the toxin, and it will bind to it and neutralize it, and preventing it from doing its job here. But you need to have this every maybe 7 to 10 years, because you need to stimulate your immune system, like the memory cell, that, to produce more antibodies. Because with time, you need to have boost to have some shot to get more antibodies, basically. And this is why when you do like hydrogen peroxide, let me ask you this, okay? Sometime if you got cut, okay, and you treat with hydrogen peroxide, right? And we know that aerobic is catalase positive, right? So they can break down hydrogen peroxide, right? So while you are treating our wound with hydrogen peroxide, we know that these bacteria will damage, they are catalase positive, and they will degrade hydrogen peroxide. So why you are using it anyway?
To kill the other germs?
What other germ?
I don't know.
Yeah, the main reason you use hydrogen peroxide is basically to kill anaerobic. So you got wound in your skin, so you treat it with hydrogen peroxide. You don't care now about the catalase positive or aerobic. What you worry about is the anaerobe. If they get inside your body, you need to prevent them from going in. So you kill them on site. They don't have catalase because they don't know what is oxygen is. The oxygen for them is so toxic. If they expose to oxygen, it will kill them instantly. Okay, they don't have, why oxygen will kill them? Because they don't have the machinery to treat, to get rid of oxygen. Okay, they don't have dismutase, or they don't have oxidase, don't have those kind of enzymes. Okay, so oxygen will kill them. So hydrogen peroxide, as soon as you add hydrogen peroxide will release oxygen, and that oxygen will kill them immediately before they get access, to your body. Deep wound, we call deep wounding. This is what you need to be scared of. If you get like deep wound, you need to be very careful, because if a deep wound get infected with all those anaerobic, that will be a huge problem. Okay, so probably I will stop here unless you have any question for me.
No, thank you, Professor.
No problem. And then we'll continue again on Friday. We'll talk a little bit about chapter 20, for gram negative.
Yes.
I do have like one question,
yes,
or what could be a question. So back to the lab, and I know we've gone past that, but I like have got mine down to what it should be.
yes.
But one important factor, and I don't want to I don't know if you want me to email it to you, but like there's one thing that it doesn't do that, and like another viable option does do. You know what I mean? Like it's offset, but for motility, I know that mine
I know what
is
you mean.
mobile.
Yeah. Well,
Yes.
do this. Do a favor for me and email me. What you're finding. What you
Okay.
think your unknown is, and look at it and see if there is any problem. For interpreting any of the biochemical testing. Then I will let you know.
Okay. Perfect.
Yeah. Just do
Thank
it.
you.
Do it. Yeah. Do it today. Okay.
Okay. Will do.
Yeah.
Thank you.
Thank you very much. Okay. So I'll see you again on Friday, guys.
All right. Thank you,
Have a
Professor.
good night.
You too.
Thank you.
Thank you very much. Bye-bye.
Bye.
Bye-bye.
Thank you, sir.
Bye-bye.